Hello everyone, welcome to Merrimack STEM Academy. Today we're going to talk about one of the most important molecular biology topics, protein structure. As you may have heard, structure leads to function, and protein structures are closely related to their functions. Before we discuss how protein structures are created, let's talk about what proteins actually do. Proteins are one of the 4 types of macromolecules, and they carry out many functions in the cell. There are transport proteins in the cell membrane. Many proteins are also involved in cell signaling pathways. Enzymes are proteins. Without enzymes, many essential chemical reactions could not occur. Proteins are also involved in structure, storage, and so much more. They are absolutely essential for many of the cell's processes. Macromolecules have structural units called monomers and polymers. Polymers are made of many small repeating units, which are monomers. For proteins, the polymer is a polypeptide, and the monomer is an amino acid. Amino acids have a few different groups, an amine group, a hydrogen group, and a carboxyl group. These 3 groups make up the backbone of the amino acid. All amino acids have the same backbone. Amino acids also have an R group. Each amino acid has a different R group. R groups can be nonpolar, polar, positively charged, or negatively charged. The chemical properties of the R group determine how the amino acid interacts with other amino acids, but more on that later. One other thing to note about amino acids, you may notice that the amine group and carboxyl group look slightly different in different drawings you see. This change occurs due to the pH level. The amino acid is essentially the same either way, so if you notice this, don't panic. There are 4 levels of protein structure. A good way to think about the 4 levels is to imagine starting zoomed in on a certain part of a protein, and then slowly zoom out to see the whole protein. If you took snapshots of the protein as you zoom out, these could represent the levels of protein structure. The first level, primary structure, is the amino acid sequence. Secondary structure is localized structure. This includes alpha helices and beta pleated sheets. Next, there is tertiary structure, which is the 3D shape and structure of the protein. Finally, there is quaternary structure, which is the 3D shape of 2 or more polypeptides. Not all proteins have quaternary structure. Now, we're going to take a closer look at each level of protein structure, starting with primary structure. Primary structure, the chain of amino acids, is formed by peptide bonds. Peptide bonds are a type of covalent bond between amino acids. The peptide bond forms between the nitrogen from the amine group and the carbon from the carboxyl group. In the amino acid sequence, the end of the sequence with the amine group and nitrogen is called the N-terminus. The C-terminus is on the end with the carboxyl group. Forming the peptide bond is a dehydration synthesis reaction. This is because when the 2 amino acids bond together, a water molecule is produced. A hydrolysis reaction is when the polypeptide is broken down into separate amino acids. It's called hydrolysis because water is used to break down the polypeptide. Secondary structure is alpha helices and beta pleated sheets. These structures form because of hydrogen bonds between the backbone of amino acids. Let's look at an alpha helix. The structure is formed when amino acids form hydrogen bonds with the amino acids below them in the spiral structure. It's important to remember that the backbone is involved with these bonds, not the R groups. Beta-pleated sheets are also formed by hydrogen bonds between the amino acid backbones. The arrangement of the amino acids and their bonds is different in beta-pleated sheets than in alpha helices, creating a different shape. Again, these hydrogen bonds involve the amino acids carboxyl and amine groups, not the R group. Now, proteins do not necessarily need to have alpha helices and beta pleated sheets in their secondary structure, but they are very common. The hydrogen bonds make alpha helices and beta pleated sheets stable. Tertiary structure, or the protein's overall 3D shape, is formed by interactions between 2 R groups. There are many different interactions that can occur between R groups. First, London dispersion forces or van der Waals forces can occur between any 2 R groups. These are very weak interactions formed when a momentary shift in electrons creates a temporary dipole moment. Hydrophobic interactions occur between nonpolar amino acids. Basically, nonpolar amino acids are hydrophobic and try to stay away from water. As a result, nonpolar amino acids may group together to avoid interacting with water. Nonpolar amino acids and hydrophobic interactions are often found on the interior middle part of the protein, rather than the protein's surface, where they would have to interact with water. Next, there are hydrogen bonds, which occur between polar R groups. Hydrogen bonds occur between the partially positive and partially negative regions of polar R groups. Ionic bonds occur between a positive R group and a negative R group. They're also called salt bridges. Finally, there are disulfide bonds. These are covalent bonds, and they only form between two cysteine amino acids. These interactions increase in strength moving from left to right across this list. The three interactions on the left are intermolecular forces rather than true chemical bonds, so they're weaker. Ionic bonds and covalent bonds are strong. Areas of the protein with lots of these weaker interactions will have a more flexible and less stable structure. If the protein needs to change shape, these interactions can be disrupted pretty easily, so the protein can change its conformation. The stronger bonds, like ionic and covalent bonds, are much harder to break. They make the protein structure more stable. For example, if the protein needs to function in high temperatures, these bonds are less likely to break under more extreme conditions. At high temperatures and extreme pH levels, the protein may denature. When a protein denatures, its tertiary structure is completely broken down, leaving an amino acid sequence with no structure. The peptide bonds between the amino acids are not affected. The primary structure will stay the same. However, the interactions between R groups and the backbone are affected. So, the secondary and tertiary structure are destroyed. Most denatured proteins will completely lose their function. Finally, there's quaternary structure, which is also formed by interactions between R groups. It involves the same interactions as tertiary structure, except the interactions occur between two or more different polypeptides. Let's review all that information. Proteins are made of amino acids. Primary structure is a protein's amino acid sequence. It is formed by peptide bonds between amino acids. Secondary structure is localized structural elements like alpha helices and beta pleated sheets. These structures are formed by hydrogen bonds between the amino acids' backbones. Tertiary structure is a protein's 3D shape. Please ignore the fact that I absolutely cannot draw a protein. Tertiary structure is formed by interactions between R groups. In order from weakest to strongest, these interactions are London dispersion forces, aka van der Waals, hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bonds. Finally, quaternary structure is the 3D shape of multiple polypeptides. Not all proteins have quaternary structure. It is formed by the same R group interactions as tertiary structure. Alright, let's put your knowledge to the test and see how much you've learned. I've drawn two amino acids, threonine and glutamate. Threonine and glutamate are found in a polypeptide and their R groups are interacting. See how many of the questions about their interactions you can answer. Pause the video here. First, what level of protein structure is this? Well, since we're looking at the interaction of two R groups in a polypeptide, this must be tertiary structure. What type of interaction is this? Let's look at the amino acid's chemistry. Threonine must be polar because it has a hydroxyl group. Glutamate is negatively charged. So, threonine's slightly positive hydrogen will interact with glutamate's negatively charged oxygen, forming a hydrogen bond. How strong is this interaction? Well, it's stronger than the weaker intermolecular forces, like London dispersion force and hydrophobic interactions, but it's weaker than bonds like ionic and covalent bonds. So it's somewhere in the middle and has medium strength. Now, there is a mutation that changes threonine to leucine. Leucine is a nonpolar amino acid. How will this change primary structure? Well, the amino acid sequence is changed, so primary structure is changed. Secondary structure involves the amino acid's backbones. The R group is changed, not the backbone, so most likely the secondary structure will be the same. What about tertiary structure? Threonine is changed to a different amino acid with different chemistry. Glutamate and leucine will interact differently. They can't form a hydrogen bond because leucine is nonpolar. Since the R group interactions are changed, the tertiary structure will most likely change as well. Since the protein structure is changed and structure determines function, the protein will probably function less efficiently. Thank you for listening. Please like this video and comment any questions you have about protein structure. Have a topic you want us to cover next? Let us know in the comments. And subscribe for more videos about chemistry, biology, and everything STEM related.